Hi guys, this is a background to the scientific rationale of the Hesperidin study that was announced earlier today. Essentially, it's a double-blind placebo-controlled study, phase 2, that will evaluate the ability of Hesperidin at the highest dose allowed in Canada to date. And it will be with a focus to reduce the symptoms and eventually reduce the likelihood of a patient to require medical assistance, and in so doing, decrease the burden on the healthcare system. In a nutshell, the pandemic is it's an uncontrolled infection that can lead to uncontrolled inflammation and cytokine storm, mainly during the adaptive immune response phase, and that's the phase that is being bypassed by vaccines that are being given right now to the most vulnerable of the people in the population. So essentially, the clinical trials and the use of Hesperidin is trying to reduce the viral load. I'll talk a bit more about that. But in addition to attempt to reduce the viral load, there's a rationale and support that it can also reduce the severity of any cytokine storm by modulating the inflammatory mediators in COVID-19 patients. So essentially, it's going to be 2 capsules given in the evening, an attempt to study the product and its impact on symptoms in COVID-19 patients, but more precisely addressing the 14 days window post onset of the disease. So we'll fly through this section. We've briefly discussed it in the highlight video that you can watch. You can also download the slide deck if you wish. So essentially, there's molecular modeling predicting that Hesperidin will bind to the region binding domain of the spike protein of the SARS-CoV-2. And in so doing, it is anticipated that it could interfere or inhibit proper binding of the spike to the human ACE2 receptors. More precisely, there are amino acids that actually form a small, shallow hydrophobic pocket that the modeling is predicting that would welcome the Hesperidin. And you can see on the bottom left-hand side, the image C, that the Hesperidin is actually smack in the middle of both the region binding domain of the ACE2 and the region domain of the spike protein. The other anticipated mechanism is the ability for Hesperidin to inhibit the replication process by inhibiting the catalytic domain of 3CL-Pro, and that was demonstrated way back with the first beta coronavirus outbreak in 2002, and then the IC50 was actually measured to be approximately 8 micromolar. So, in a nutshell, the catalytic domain of the 3CL-Pro has remained constant between the 2002 outbreak and the pandemic that we're living through right now. So the likelihood of a change in a potency has been significantly reduced as the catalytic domain has remained consistent. Further than that, is that there are now ample reports by more than one group, all essentially confirming in their own modeling way the potential of Hesperidin to block the 3CL-Pro. So when you resume or not resume, but as a sort of a bottom line here, the hesperidin is anticipated to inhibit the entry of the spike protein by binding to the shallow hydrophobic pocket of the region of the binding domain, and also to potentially bind to the 3CL pro catalytic domain um, with an IC50 of about 8.3 micromolar. So in the second part, it's a description really of the way that we educated ourselves and going about the literature that we wanted to find. I don't want to go through all of this, but essentially it's the path post-infection, because once the cilia of the trachea, the bronchi are infected, these cilia's purposes are to move mucus out of the lungs. But if they're infected, they can't do their work, and the mucus particles and the virus become more stagnant. And that leads to a first inflammation response. And within the inflammation response, inflammatory mediators like cytokines. And the cytokines are very important because when, with regards to influenza, and it theoretically should be very similar to the COVID-19, IL-1 and IL-6 are key in inducing fever. And the inducement of fever has been referred to by virtually every group as the day one of the onset of the disease. And it shouldn't be overly surprising because fever has been observed even in controlled situations with vaccines. There are various types of vaccines that can elicit fever. And that fever typically occurs within the first 24 hours to 2 days post-immunization. So as you go through this schematics, the time of onset, when you stated at 1, by the 7th day, you may require medical hospitalization. And fever has been reported time and time again over the last year as being one of the most important marker of the onset of the disease. Not the only one of the very important one. And when we look at fever and fever trend between survivors and the non-survivors, and the clinical course for the ones who have survived on the left-hand side, the observation is that after the lag time period, once the body and the B cell are able to generate neutralizing antibody against the coronavirus, typically in survivors, fevers goes down. But unfortunately, in the patients that didn't survive, there's a large number of them that the fever remain constant after those 7 days. And you can see that death starts to increase as days move forward. And so one of the interesting things about the temporal dynamics in the viral shedding is by the day of onset, typically the viral shedding is in a declining phase. It's still present, but it starts to decline based on this paper. And so the reason why masks are important and the reason why you don't exhibit symptoms until typically the viral shedding has occurred. And when folks are not careful and don't wear a mask and safe distance, then the likelihood of transmitting the disease is a lot higher before you get your first day of onset of the disease of COVID-19, essentially. And that's been observed from a theoretical to a practical. And you can see here on the left-hand side that in the first 7 days, typically antibodies capable of neutralizing the coronavirus or absent from plasma of folks. And COVID-19 patients don't respond all the same way. The infection level is different. And even with a vaccine, for example, that bypassed this lag time, I mean, that's the purpose of the vaccine. The response will vary from one patient to another. When you combine all that information inside of a sort of a attempted window between the progression of the lag time and the inflammation, the observation of neutralizing antibody against the coronavirus, and the viral dynamics, it provides with it an overview of the objectives of the study with Hesperidin in trying to both reduce the viral load and attempt to modulate the inflammatory mediators. Now, the basis of the research essentially into which inflammatory mediator is key or that should be monitored or modulated was guided by the Reprogram Consortium, which is a nonprofit group. And so I'll go fairly rapidly through this segment. Again, you can download the PowerPoint and you can have a look. But essentially, we developed this based on those recommendations from that particular group. And so they recommended, the cytokines leading to cytokine storms. They recommended also, TNF alpha and the likes, and also here all the reactive oxygen species, the ROS, that play a key role, and also the influences of the ROS on NF-kappa B and NLRP3, and we'll get into a bit more of those a little later down the road, in addition to the superoxide dismutase that play a key role. So we educated ourselves and we drove conclusions or assumptions from what was published to understand what would be the key inflammatory mediators and their relationship with Hesperidin. For example, is there any literature about Hesperidin influencing positively those key inflammatory mediators in COVID-19 patients? That's the foundation of what is presented here. And so within that paper, they also suggest all the various agents or strategies to try and modulate, and there's many of them. And so this is a massive task to try and infuse or provide all of these therapeutics or potential therapeutics to a patient that is either in an ICU unit or in a ward. And so to close off here, the consortium presented the importance of the JAK-STAT signaling pathway, and especially the IL-6 receptor family. And so we started to gather additional information based on the patients themselves on the left-hand side, or the progression that was observed early on in the patients, mainly in Wuhan. And we've also looked into the crosstalk between coagulation and inflammation. And you can download the papers, all the papers are cited here, and have a look at this crosstalk that does exist between essentially the two systems. And so that allowed us to generate a list, right? A shopping list of with the big question, can Hesperidin influence any of these inflammatory mediators? And so in the third part, we'll go over just a few papers. There are many papers out there, and we selected the 7 of them for essentially three key organs. The first one is the heart. Of course, we're very happy to do this clinical program with the Montreal Heart Institute. And you can see here that Hesperidin, within just a single study, was able to influence many of the key inflammatory mediators in a acute myocardial infarction model. Furthermore, within that um, paper, you could see that Hesperidin was able also to influence the key cardiac troponin I in those models, along with a cascade leading to that decrease of the cardiac troponin I. And so we were able, within two papers, to check box certain inflammatory mediators, which, according to literature in animal models, was influenced positively. And then we moved on to the lungs. And again, in the lungs, within three papers, there are reported dose response of Hesperidin in various models, like this one, which is an acute ventilator-induced acute lung injury, which was observed significantly in the early onset of the pandemic, and COVID-19 patients needed to be placed on ventilators, combined with the report of endotoxin-induced acute lung injuries, what again influencing positively many of the key cytokine inflammatory mediators, and coupled with a publication from just a couple of years ago on sepsis-induced lung injury, and especially the toll-like receptor 4 influence and the MyD88 influence by Hesperidin in a dose response fashion. So that allowed us to check box another series of inflammatory mediators. Some of them are redundant, like IL-6, for example. And we moved on to the neuronal aspects or the potential influence of Hesperidin on the CNS. And here again, you can see very recently published in 2020, dose response, and you can see key inflammatory mediators being moved positively across the board in a dose-dependent fashion in a sevoflurane anesthetized rat model. In addition to an extensive review published in 2019, where there are 19 different types, this is just a highlight here, but there are 19 different types of animal models where Hesperidin was reported to provide benefits in the CNS of those models. So consistently, various models all presenting evidence of benefit of Hesperidin. So that allowed us to check box another series of inflammatory mediators. But essentially, we were able to capture within 7 papers, and there's more than 80 of them, but within 7 papers, a nice distribution, consistent within across organs, the 3 key organs anyway, for COVID-19 patients. And we were able to essentially, just the schematics, say it looks like a complicated, but it's not really complicated. The main distinction is the Hesperidin clinical trial is to an attempt to prevent patients from requiring hospitalization by decreasing the viral response and an inflammatory response. Whereas remdesivir that was approved initially was for patients that were already hospitalized. And so I'll let you go through this, but essentially it's the support of why those 14 days are important, why the day of onset is important, and that the clock starts when you have symptoms. If you don't have symptoms, you're asymptomatic, and that's great because you're not going to require hospitalization. You're asymptomatic. So all of this is also driven by Hesperidin being a bioflavonoid, and bioflavonoids structures are influenced by hydrolysis, by gut microbiota. And so we looked at a lot of literature on pharmacokinetic profiles of hesperidin in humans, and we were able to generate a first dose, as this is a phase 2, a first dose, which is the maximum dose allowed by the health authorities in Canada to date. That may change, but to date, that's the highest dose. And that's a dose that is significantly higher than any daily intake of hesperidins from different citrus sources. So in a nutshell, this is the program at the Montreal Heart Institute. At the bottom, you can see that it's also important to decrease binding and the replication. But it is the bulk of the scientific rationale behind the hesperidin study that was announced earlier today. That was made some time ago, but I think it's still valid because what is observed is that the flu and many of the infections, resolution of the infection via the antibody is the normal path forward, but often uncontrolled inflammation is what leads patients to require hospitalization, and flu is no exception. And so just one slide here to present the ability of hesperidin to attenuate the influenza A virus, and that was done in 2018, through essentially the anti-inflammatory effect or the modulation of inflammatory mediators, as we talked about in the previous slides. And you can have a look at this, the reference is there, the paper is available. And so we would like to thank you. Thanks to Montreal Heart Institute, and especially all the healthcare workers around the world on the front lines of this coronavirus pandemic. A heartfelt thank you. Stay safe, and you can download the presentation, which is right next to the video.